All right, here we are, folks. It is Monday, the end of November, the 30th, and we are working on trying to finish up our cells unit. We're looking at our cells unit test next week on Wednesday, so that is going to be somewhere in the neighborhood of about December 10th. Just a heads up so everybody can start to prepare. We've spent a lot of time talking about a lot of things, especially talking about passive and active transport. Make sure that you know the difference. But today, for this week, what we're going to be doing now is we're going to be talking more about the organelles. Last week, I gave you guys a project to go through and pick out 5 organelles and to create some form of presentation, giving me their name, what they look like, and what they do, and to put in something creative like a magazine or a song, something like that. So I hope everybody had some fun with that. That also means that some of the stuff we're going to look at today should be review, because you should already know several of these. But let's get started. I don't want to waste too much time at all because, there's just so much for us to do this wonderful, wonderful week. So let's start off with, where is it? Nope, there's the right button. All right, so starting off, in Schoology, you will see we are still in Unit 3 for this whole week. And inside Unit 3, we're now into Week 4, because today is the 30th of November, and Friday is the 4th of December. Now, if you take a look, what I've done this time is I've tried to clarify things a little bit more for you. So the cells, I'm literally telling you exactly what slides we will be working with. And for the assignment, I'm giving you the days that we'll be working on this. In this case, Monday/Tuesday, depending on when your lab bell is. So, and then of course our turn-in. So this will handle our first 2 days of this week. And let's dive right into the cells, because we've got a bunch of organelles to go over. If I move too quickly for you, please feel free. Pause the video, get the information that you need to, and then move on to the next. So we are starting on slide number 48. I had that listed. All right, so remember, organelles are going to be those smaller parts that have a job to do. We're going to start with the cytoplasm. And all you really need to know about this is that the cytoplasm is the filling inside the cell. So all the stuff, all the organelles, kind of float around in this jelly-like substance that just keeps them there. It basically maintains a home address for them. The nucleus. All right, the nucleus has 2 very important jobs. Number 1, it controls the cell. It's the boss. It's the person in charge, right? It's the CEO. If anything in the cell wants to do anything, they got to get permission from the nucleus. And that is because it contains the genetic information. The genetic information is like the booklet that tells the cell how to do all the things that it needs to do. And so he who controls the information controls the cell, because the nucleus has the DNA information within it. It is therefore the boss. Other parts of the cell do not know how to carry out their jobs. They simply have the machinery required to. Good memes. The nucleolus. Now, the nucleolus exists inside the nucleus, and it's not really an organelle itself. It's kind of just a splotchy goop of slightly more like condensed stuff. So it's a little bit stickier and more thick than the rest of the nucleus. But it makes the ribosomes. It makes the little pieces, and then those ribosomes get spit out of the nucleus into the cell to do their job. which means, well, what does a ribosome do? Well, ribosomes make protein. And that's the key. They make protein. So all of our muscle fibers are created by ribosomes. What else? What other important biological molecules do we know of that are made out of protein? They're part of the protein group. Very important to keep alive are enzymes. Enzymes are proteins too. So these ribosomes are building everything that we are, our physical form, our protein structure, and they're also going to be building all of those control chemicals within your body. Huge. Unbelievable that it comes from the smallest organelle that we've found. Now, the endoplasmic reticulum. It sounds very impressive. If you write down capital E, capital R on any test, quiz, anything in biology, anybody grading it will know that you mean endoplasmic reticulum. Nobody will mistake it for emergency room. So don't worry about needing to spell this out. If you can just say ER and remember it as the ER, you'll be fine. And both of our ERs, they do have slightly different jobs, but we are going to simplify things down to understanding that they both make lipids, namely the membrane lipids, that phospholipid bilayer. Now, there are two types, our rough and smooth. The only difference is that the rough ER has ribosomes on it. So it gives it this kind of studded image. A lot of people will say it's like a dirt road that has little rocks and pebbles in it, aka rough. Whereas the smooth ER does not have any of these bumps, none of the rocks. So it's like blacktop. Then we have the Golgis. Now, the Golgis function as a mail system for your cell, meaning they need to move material and package it. That is what they do. The way they do that is I've colored these yellow circles up top, right? And they are moving into the layers. And as they pass through each layer of the Golgi, what's going to happen is they get wrapped in another layer of lipids and proteins. And that wrapping provides the packaging and the stamp to get them wherever it is they need to go. Now, that's done technically by adding lipids and carbs to them. But if you say that it packages and ships, you'll be fine. The lysosome. This is a very small organelle. You will not be able to identify them individually from most diagrams. You won't be able to tell the difference between them and a vacuole. They're just these little tiny, seemingly empty circles. But their job is to break down old and damaged organelles. So if you imagine a factory, right? A factory's got a whole lot of different machinery in that. They're all making different things. Eventually, some of that machinery will wear out. Your cells are no different. Our organelles do wear out over time, and they need to be replaced. The lysosome's job is to break them down so we can recycle those materials and build a new one. And they do this by using enzymes. This whole bubble is full of protein enzymes, biological catalysts. Remember, they control reactions. Hydrolysis breaks things down, and so they can break down any part of the cell they're in. This should look familiar. Then we have the second one. Now, in a plant cell, the vacuole is really easy to identify. It's huge. In plants, the vacuole is enormous. In animals, it's a lot harder to tell. If you take a look at the diagram behind me, this is a vacuole, and those are lysosomes. The difference is very minimal. And what they are is this is a storage bag. It's just a place to put stuff. Maybe it's food. Maybe it's water. Maybe it's waste. They just take stuff and they store them into our vacuoles. We have chloroplast, super important to plants and for life on this planet because this is where sunlight is turned into food. Now, we do not have, there go the lights. We do not have any examples of this in animals except for one single-celled organism called a euglena. And if you remember, turning sunlight into food is called photosynthesis. We'll be working with that a lot later this year. And here is photosynthesis. Now, for right now, don't worry about getting this information quite yet. We're going to see this and work on this over and over and over again. But I wanted to remind everybody of what it looks like. And its goal is to take water and carbon dioxide from the air and turn it into sugar, C6H12O6, and oxygen. Really useful for us. Now, the mitochondria, the powerhouse of the cell. All right, so remember, the mitochondria is the energy factory. This makes all the energy that a cell is going to need and use. So this takes those food molecules that we eat and it breaks them down and releases the energy from them. And you're going to find them in different amounts depending on the tissue of your body. So for instance, the heart and muscle tissue are just full of mitochondria, whereas something like your eyes have very, very few comparatively. Now think about that. That's on purpose. There's a reason for this difference. And we finish finally with the cytoskeleton. Now, this is generally not considered an organelle, but it does play a very important role. So what it does is it creates the framework for the cell. It creates the scaffolding that's going to hold that cell in shape. It also makes all the little roads and pathways inside the cell for things to move. If you take a look at these pictures over here, right? What they did was they took the 3 different types of fibers that make up the cytoskeleton and they dyed them different colors. So the microtubules are red, the actin filaments are green, and the intermediate filaments are blue. And so you can see where they are in their greatest concentrations within the cell. So we see that the microtubules really form the outer layer, and that gives our cells the shape that they have. The actin filaments are the road networks throughout the interior. And the intermediary filaments, the blue, those are really just in the largest organelles. The nucleus here. These are mitochondria around the sides. So that's really where we find them. Now, our lab today is going to be based around these organelles and what they do. So I hope to see everybody there. I hope that this made things a little bit more clear if you had issues with the project. And I hope to see you all in class. Have a great day.